Thank you all for sticking with this. It's tough to be in the graveyard shift, but hopefully this will be a nice sort of fitting end to the conference. And I'm one of these people that's been very fortunate to have seen a whole evolution of non-invasive prenatal screening and testing really over the last 20 to 30 years. And I feel very strongly that the story won't be complete until we have a fetal treatment option for pregnant women whose fetuses have been diagnosed with Down syndrome. I have no financial conflicts of interest with regard to this presentation, although Illumina did pay for part of the work, but it's a sponsored research grant that's administered totally through Tufts Medical Center. Now, some of you may have heard my talk a year ago at the meeting at Brisbane, and I just want to tell you that a lot has happened in the past year. So, for example, there was the very first Trisomy 21 Research Society meeting held in Paris about a month ago, and during that meeting, there was a historic entire first session in which there were 6 speakers shown here who were all discussing different aspects of prenatal treatment to improve neurocognition in Down syndrome. So it was one of these meetings where you could really feel that the paradigm was changing. Now, the vision is that prenatal diagnosis would create an opportunity for fetal treatment. And I don't know if Aubrey Molunsky is still here, but I would like to quote him again on just because you can doesn't mean you should. But I do feel that we should try with regard to the research. And this whole concept has really evolved in the past 3 years. So while there have been many trials looking at treatment for Down syndrome, for individuals with Down syndrome in childhood and adulthood, the concept of treating fetuses to positively impact brain development and to improve neurocognition has really been something that has evolved over the last 3 years. And in 2012, I wrote about this potential opportunity, anticipating that sequencing of cell-free DNA in the maternal plasma would become quite popular. And the hope would be, following a confirmatory diagnosis, that women, when they received post-test counseling, would be given a third option. And that would be that eventually there would be some sort of treatment that the woman could accept. And as someone who has been working in reproductive genetics, although I am a pediatrician by training, I have seen consistently that when pregnant women are given an option of some sort of treatment, whether it's surgical or medical for their fetus, it's something that they're very interested in. So again, I feel very strongly that we should try to pursue this and do responsible research. So the idea is that we would eventually give a drug to the mother as treatment for her fetus. And you heard in Dr. Carr's talk about some of the ethical considerations. You know, who's the patient, and is it right to give the mother something for the baby? And that's very relevant for this work as well. But not treating prenatally is a lost opportunity to improve neurogenesis and positively impact upon brain development and brain connectivity. So one of the first things we've been doing is to really pull together a more comprehensive picture of brain pathology in human fetuses with Down syndrome to show the world that there is an opportunity. And one of the most striking things about the meeting in Paris was that one of the presenters showed that there's actually evidence of Alzheimer's pathology in fetal brains of fetuses with Down syndrome. So you might not only be able to just improve neurocognition, but perhaps prevent Alzheimer's and dementia. So in the talk, I'm going to talk about human fetal pathology, then some of the considerations with mouse models, then approaches to prenatal treatment in mouse models, and finishing up with preparations for a human clinical trial treating pregnant women carrying fetuses that are known to have trisomy 21. So is there a phenotype in human fetuses with trisomy 21? I've already told you, yes, there is. It's most apparent in the second trimester. I'm not aware of studies that have been done in the first trimester, But certainly by the second trimester, by ultrasound examination, you can see anatomical differences primarily in the cerebellum and the frontal lobe. And what you see characteristically is a decreased frontothalamic distance and a decreased transcerebellar diameter. And if you look at postmortem analyses, you can see that brachycephaly and microcephaly is developing by 15 weeks gestational age. So if you were to diagnose the fetus before that time and initiate therapy, presumably you would begin to have a positive impact. But these studies show that brain weight, overall brain weight, decreases by the second trimester, and in term fetuses, the cerebellar volume is 79% of age-matched control fetuses. Not only do you see a gross difference in brain size, but you see histologic differences. So you see in the second trimester, this is work of Guidi et al. From Bologna, and you see that affected fetuses have decreased neurons in the hippocampus due to decreased proliferation and increased cell death. And this is, these are slides that are stained with Ki67, which is a marker of cell proliferation. And you can see here, this is the euploid fetus, and there are a lot of cells, a lot of brown-stained cells. And in the age-matched fetus with Down syndrome in the brain, there are fewer cells and fewer dividing cells. And you see that as well. It's highly statistically significant. And this is work that Tomo Tarui from our group, who presented the other day, who's been looking at living fetuses by MRI, and he has shown with motion correction studies that the cerebellum in various various metrics, that the cerebellum is consistently smaller in fetuses that are affected with Down syndrome. We've also been doing transcriptomic work. This is work that's from a number of people in the laboratory, including Lisa Hoy, who may be in the room. I see Keiko Koide is in the room. And we have been looking at cell-free RNA in amniotic fluid. And what our group has shown is that the cell-free RNA in the amniotic fluid actually contains transcripts that map specifically to the brain, so that you can get information on the fetal brain from this amniotic fluid cell-free RNA, and that is real-time gene expression data from living fetuses. There are a number of other organs that you see here we also get information from. And using that information, if you look at fetuses with various chromosome abnormalities and compare them to euploid fetuses that are matched for sex and gestational age, you get these very characteristic gene expression signatures. So we've summarized this work in a paper that's gone online recently, but here we're focusing on fetuses with trisomy 21, and they are characterized by having massive amounts of oxidative stress, abnormalities in cell proliferation. You already see signals of neuroinflammation at 15, 16 weeks of gestation, as well as other abnormalities including ion transport and G protein signaling. Contrast that to fetuses with Turner syndrome, which we've also studied. And adult women with Turner syndrome have significant problems with hypercholesterolemia. You get aortic abnormalities, and the fascinating thing is all of that is there prenatally as well. So despite the fact that this is cell-free RNA, each of these transcriptomes gives you a completely different picture that is very consistent with postnatal pathophysiology. Now, why is oxidative stress the hallmark of trisomy 21? Well, in this study by Gimeno et al., they took fibroblasts from human fetuses with trisomy 21 and showed that the ratio of copper to zinc superoxide dismutase activity to catalase and glutathione peroxidase activity is increased, and this produces increased hydrogen peroxide. This results in oxidative stress and induces mitochondrial dysfunction and starts this whole very abnormal pathway. In addition, these fibroblasts from fetuses with Down syndrome have very short telomeres, an increased expression of DSCR1, Down Syndrome Critical Region 1, also known as ARCAN1, which codes for a protein that suppresses VEGF and decreases cell proliferation. So in every which way that we look at these human fetuses, we see oxidative stress, we see abnormalities in cell cycle, we see abnormalities in cell proliferation. And even directors of cytogenetics labs will tell you that quite often the amniocytes from fetuses with trisomy 21, they almost know ahead of time because they're not growing well in the lab. In general, because of the short telomeres, the cells are acting like they are from a much older person. So let's just move quickly to mouse models. Fetal Gedge, also from our group, presented the other day about the TS1 CJE mouse model that we use preferentially. There's quite a bit of discussion amongst the researchers in this area about the best mouse model to use. This is human Homo sapiens chromosome 21, and the majority of chromosome 21 in the human is orthologous to mouse chromosome 16, with a little bit of 17 and 10. The three major mouse models that are currently used are orthologous and contain sections of the chromosome 16 genes. They are the DP16-1YEY, the TS65DN, and TS1CJE. TS65DN is the oldest one that's been used. There's a lot of publications on this mouse. It contains 128 genes from the mouse chromosome 16, but it also contains some genes that are not related to human Down syndrome. The reason why we don't want to use TS65DN is that the males are sterile. So that means to pass the extra chromosome to the mouse pups, we would have to pass it through the mother. And you'll see when I show you some of the other experiments that people have done, they've used the TS65DN. But we feel that this is a confounder because it's not a normal intrauterine environment. These moms have abnormal reproductive performances, and we're doing a lot of behavioral testing. And in this situation, if the mother is affected with Down syndrome, she will not necessarily teach the pups what they need to know. So it's very hard to evaluate the effects of treatment if the mother is confounded in the experiment. So there was a lot of excitement about the DP16-1YEY, which was created in 2007. There's relatively little known about these mice. The males are fertile. It does not contain any genes unrelated to what are on 16. But the problem is, as we found out recently, there is no prenatal brain phenotype in this mouse. So therefore, we've decided to work with the TS1 CJE mouse, which is sort of like, one is too hot, one is too cold, one in the middle is just right. It does have the majority of the genes, but not all of the genes in that chromosome 16 section. It has a few genes, unrelated genes from chromosome 12. The males are fertile. These mice do have oxidative stress in their brains, and there is a prenatal and an adult phenotype. And Fetal has recently published an extensive series of embryonic and neonatal endpoints, so that we were very interested in objectively understanding exactly what is abnormal about these mice so that then we can measure the effects of treatment. And that was published recently. It's online in the American Journal of Medical Genetics. Now, this is a picture that I took in the mouse facility. It shows you that it's really impossible to be blinded for these experiments because these are all litter mate controls. So these mice are all the same age, and yet you can see these two are much smaller. Now, we don't know at this time if they are TS1 CGE, but we could, because they are all blinded at the time they're being tested. But just looking at them, we pretty much know that they are TS1 CJE because they're so much smaller. And they also have little white spots on their back, which is consistent with a younger appearance in a particular C57 black 6 background. So these mice are not only small, but they have a general delay in development of their fur. Fetal has studied the functional differences in the embryonic brains of these mice, and they have almost all the things that we're interested in. They have decreased cell proliferation, they've got increased cell death, they do have oxidative stress. Interestingly, we've shown that both the mice, and now it's increasingly recognized in humans, that there is an abnormality in smell in Down syndrome, and smell is an important part of memory, and learning and memory is what we're trying to improve. Sorry, this is a similar slide. I do want to show you just quickly some of the behavioral abnormalities. We have a whole series of tests that we perform between days 3 and 21 of postnatal life. And so what I'm going to show you is the cliff aversion test here. This is the normal mouse now, the wild type mouse. They are blinded. They're literally blind at this stage of age, and they should sort of feel that they're at the edge of a cliff and then begin to pull back. So, sorry. Just lost the... So this mouse recognizes that he's at the edge of the cliff and he's turning around. That's the wild type mouse. Okay? And now you're going to see the TS1 CJE mouse. We never let them fall down the cliff, don't worry. But you can see that that mouse doesn't really react or is so weak in the four limbs that he can't pull back. So what are the current approaches that are being taken for prenatal treatment? There have been some experiments. All of these experiments have been done in the last 3 years. And I'm going to briefly review for you the neuropeptides, NAP-SAL. I'm going to review for you epigallocatechine gallate, EGCG, maternal choline supplementation, and then fluoxetine. So NAP and SAL, this is work done in Kathy Spong's laboratory here at NIH. Really comes from some of their related work in fetal alcohol syndrome. But it turns out there are high levels of vasoactive intestinal peptide mRNA in the brains of TS65DN mice, but there is a defect in the signal transduction mechanisms of the receptor. So Inserti et al. In this particular study gave the neuropeptides NAP and SAL, which are abbreviations for very long compounds, or saline to pregnant TS65DN mice, and the treated pups had improved spatial learning as adults. And the only way they were tested was with the Morris water maze. And since Kathy has now become deputy director of the National Institute of Child Health and Human Development, her lab, she doesn't have a lab anymore, so she's not pursuing this work, unfortunately. The same year, a group at Cornell in Ithaca, New York, published on maternal dietary choline supplementation. And choline is an essential nutrient during fetal development due to its role in acetylcholine production. It's also important for cell membrane formation and epigenetic regulation. And the maternal choline supplementation produced dramatic improvement in spatial cognition, attention, and emotional regulation in older TS65DN offspring. And this correlated with hippocampal neurogenesis and increased density and size of basal forebrain cholinergic neurons. Now, as far as EGCG, this is work that actually Fetal Gedge did in Paris, I should say 2009. And this is contrasting what I would call the single gene approach to something that I'll be talking to you about in our laboratory, which is more of a systems biology approach. But this work is aimed towards treating DYRK1A dosing. And DYRK1A is a master kinase in the Down syndrome critical region that controls excitation, inhibition, balance in the brain. And with a variety of mouse models, they've shown that the dose of DYRK1A affects brain growth, formation of dendritic trees, learning, and memory. Now, EGCG, which is a natural polyphenol, it's a major ingredient in green tea, inhibits expression of the gene DYRK1A. And we heard at this Paris conference that there is a trial going on in various transgenic mice looking at treatment of DYRK1A expression with EGCG prenatally. And just with preliminary data, they've shown that offspring of prenatally treated dams show increased neuronal density and improved excitation, inhibition, balance again. But unfortunately, that's not published yet. Now, lastly, fluoxetine has been published, and this again is the work of Renata Bartasaghi's group in Bologna. And it relates to the fact that the serotonergic system is important for neurogenesis and dendritic development. And this particular system is altered in individuals with Down syndrome. This group gave fluoxetine, also known as Prozac, to TS65DN dams from day E10 of pregnancy. So a mouse pregnancy is about 18 or 19 days, so sort of halfway through the pregnancy, up until delivery to inhibit serotonin reuptake. And the treatment, which they showed in great detail in this publication, restored overall cellularity and connectivity in multiple locations in the brain. And these effects persisted long term. So they had one group of mice that they studied right after delivery, and they had another group of mice that they studied 45 days later, although they were only treated through the pregnancy. And even the long-term mice had improved cognitive performance, even though there wasn't ongoing treatment. Our group, which includes Jeroen Pennings, who's also here in the audience, is taking a pathway approach, not a single gene approach. You can see DYRK1A is right here. So these are all candidate genes to treat that are somehow affected in individuals or mice with models of Down syndrome. And the pathway approach does identify all of the things that seem to come up repeatedly in terms of what is different in Down syndrome compared to age-matched controls. And you see response to oxidative stress is different, apoptosis is different, neurodegenerative disorders, neurogenesis. These are all the areas that we need to treat. So we use the connectivity map, which is a publicly available database that allows us to upload all of the differentially regulated genes. So we can do this in the human. Jeroen Pennings has translated it to the mouse for us, so we can upload the differentially regulated genes in the mouse brains, and then we can identify drugs that are FDA approved by the US Food and Drug Administration, and these drugs reverse the entire picture of the dysregulated genes. And so it produces a list of candidate drugs, and then we review them for safety in various databases, because some of the drugs are approved, but they are not drugs that you'd want to give during pregnancy. And I like to think about the connectivity map as a match.com, meaning opposites attract. So what you're trying to do is reverse the abnormal gene regulation. So the first test drug that we've pretty much run through, we're now evaluating the data, is apigenin, which is a naturally occurring flavonoid. It's an antioxidant. It inhibits oxidative stress and apoptosis. It's present in leafy green vegetables and citrus fruits. In other studies, it's been shown to cross the blood-brain barrier. It's been used in various clinical studies, including Alzheimer's trials in humans, as well as prostate cancer trials in humans. And so we have undertaken a very extensive series of experiments where we are comparing wild-type black 6 female mice mated with the TS1CG male, because as I said, they're fertile. Half the mice are untreated, half the mice are treated. Within each gestation, approximately half the pups inherit the extra chromosome. So you actually have littermate controls who do not have the extra chromosome, and they've been exposed to the same in utero environment. So it's a very good control system. And we are comparing gene expression in embryos. We're looking at neonatal behavior. We're looking at brain histology. And in 8 to 10 week adults, we're also looking at advanced neurocognitive behavior. This is work that's not completed, so I can't give you a full story, but it does look like there is a positive effect. with regard to the embryonic gene expression. The apigenin normalizes gene expression in many of the genes, but not all of the genes. It would be unrealistic to expect that one magic bullet would create, just completely normalize everything. But in general, the effects are more positive. Some effects are neutral. In the neonate, there's no change in growth or appearance. So the treated neonates still look small. You may have seen Millie Ferez's poster at lunchtime in which she's been looking at the cardiac defects. It turns out these mice do have cardiac defects, and so the treated mice have not been evaluated yet to see whether the amount of cardiac defects remains the same. But the important thing is the treated mice do take less time to achieve the developmental milestones. So what we see consistently is even that little guy who looked like he was falling off the cliff, eventually he will figure out how to achieve that goal. And so what we see characteristically in the TS1 CGA mice is that there is a delay in their development, but the treated mice tend to achieve their goals much faster than the untreated mice. And in the adults, we do see improvements in certain behavioral tests. The open field test relates to exploration of a cube. There's a typical type of behavior that's normal. In looking at differences between males and females, we do see that the apigenin does improve things for the male adult mice much more significantly than the females, although the females do have a therapeutic effect. And we also see some improvement in memory in the fear conditioning test, where the mice have to remember that they've been in a very stressful situation previously where they've gotten a shock, and the normal reaction is to freeze. So there is some improvement there. And the brain histology is as yet unknown. We know that in the untreated TS1 CGA mice, we do see abnormal accumulation of cells in a particular part of the brain called the median ganglionic eminence, and we're waiting to see what the effects of treatment are on the histology. So what are the preparation steps for a human clinical trial? Well, the vision is to get the prenatal diagnosis, give the woman the option. If she wishes to take the drug, she takes the drug for the fetal benefit. We then will evaluate the child, and the ultimate goal, this happens to be the son of my mentor at Stanford University, the late Leonard Herzenberg. This is his son, Michael Herzenberg, who was the inspiration really for my entire career in prenatal diagnosis. But you can see that Michael, as an adult, has severe microcephaly. And the hypothesis is that the oxidative stress is essentially destroying a population of neural stem cell progenitor cells, and those cells never come back. And that is the reason why adults who have Down syndrome have microcephaly. So in the human, we have a lot of considerations, much more complicated than treating mice. We need to identify compounds that are safe for pregnant women and their fetuses, that cross the placenta. We need to understand dosing. We need to show clinical improvement and lack of harm. And just because we're treating during pregnancy doesn't mean we're not going to treat after pregnancy, and it might be that one drug is safer and more appropriate to give during pregnancy and something else might be safer to give later on. But I want to stress upon you that we don't have a lot of time to wait. Almost weekly, if not every other week, I get emails like this. So, the world is a different place. Everything is open, everything is on the internet. Anybody searching for prenatal treatment for Down syndrome finds our publications, and here's just an email from a woman who's telling me, this is from April of this year, I'm currently 19 weeks pregnant. I've learned that our son has trisomy 21. I'm a physician assistant. I want you to now here she is. She's writing me an email. I don't know who she is. I realize you can't recommend anything not tested and approved for humans, but I'm wondering if you know of any modalities that might help or at least wouldn't hurt to try. I would love to feel like I'm doing something to help him. So these are the kinds of people who are actually self-medicating, and that's very worrisome. When we were in Paris, there was an exhibit from this company that's making a product called Font Up, which is a preparation of EGCG. It's actually in a little chocolate shake. They were giving away packets of it. I should have taken it, but I didn't. I was afraid I was going to get stopped in customs or something. Like, what is this? I don't know. But it's, it's being marketed. You can see here, so here's a delightful-looking young man who has Down syndrome, and it's implying that this is going to help him with self-sufficiency and competence in daily life. It's going to increase his learning. It's going to increase his social functionality. It's going to increase his cognitive performance. It's a non-prescription green tea supplement. It's already on the market in Europe for children who have Down syndrome. There's no doubt in my mind that pregnant women are probably taking it too. So it's, as I said, the marketing is suggesting that it's really going to turn things around for your child. On the back, which I didn't copy for you, but there's a whole set of sort of pseudo-scientific graphs and all of that. So again, things are moving very quickly. Oops, sorry. So my concerns are, what are the active compounds in green tea? One of the scientists at the meeting, Randall Roper, told us about the fact that he measured different products sold as green tea, and the amount of EGCG in each of them was quite different, and each one of them had different additional compounds in it. So the reproducibility of different preparations is very questionable. And also very little is known about the transport of EGCG across the placenta. But again, people can get it and self-medicate. All right. Here's another thing that's going on. This is a crowd-funded study to treat Down syndrome prenatally. Look at this guy. This guy is Paul Watson. I looked him up. I think he's a pilot for Southwestern Airlines, and he has raised $85,303 as of early July to fund a prenatal clinical trial of fluoxetine. Okay, fluoxetine is Prozac, and of course Prozac is given to pregnant women with depression. But this is Prozac that's going to be given to women without depression who have fetuses with Down syndrome. It is an IRB-approved study. I don't know any of these people. I don't know anything about the study. But if you read what it says on the internet, there is nothing at all about the possibility of birth defects associated with fluoxetine, and I'll get into that in a moment. But it looks like they're going to recruit 21 pregnant women at UT Southwestern. If anybody's here from UT Southwestern, please let me know if you know anything about this study. Now, fluoxetine and birth defects is a very hot topic. There have been multiple prior meta-analyses and systematic reviews. A lot of controversy about safety, and that's for women who have depression who need to take fluoxetine. There was a paper published in the BMJ last week that looked at over 38,000 pregnant women, taking note of over 30 major birth defects, and showed very conclusively that fluoxetine exposure was associated with ventricular septal defects, right ventricular outflow obstruction defects, and craniosynostosis. This study did not look at the brain, and clearly it has effects on the brain, both on the mother and the baby. So in our laboratory, we are really trying to take this more cautious approach, but a rational approach, and we have a list of other drugs that have been identified by the connectivity map to reverse the abnormal prenatal transcriptome in trisomy 21. And we're going to compare the effects of prenatal treatment alone versus postnatal and prenatal treatment in the TS1 CJE model. And then we're simultaneously preparing for a human prenatal clinical trial of whatever the best and safest drug is. And part of Tomo's work is really to establish baseline untreated parameters so that then we can do MRIs, for example, on women undergoing treatment to see if we already see improvement in cerebellar growth. We also need to demonstrate that the drugs cross the placenta and do no harm, and we've already had a lot of interest from various recruitment sites, not only in the United States, but in Chile, Japan, Qatar, and Ireland. So in summary, developmental brain abnormalities exist in human fetuses with trisomy 21 and also in a variety of mouse fetuses with orthologous genes, and treating mice prenatally appears to reverse defects such as reduced neurogenesis. And these effects, at least in some of the studies, do appear to be long-term and may be permanent. There's no mouse model that perfectly replicates the human condition of trisomy 21, so it's only a model, and eventually we want to get back to the humans. I do want to communicate today, and I hope I have, that research in this area is very active and increasing. However, funding for this research is a challenge, and so it's no surprise to me that somebody did a crowdfunding attempt, because NIH is funding very little in this area. A human prenatal clinical trial is already ongoing, and likely there will be more in the future, but safety is paramount. But lastly, I want to leave you with the message that prenatal diagnosis really should be considered an opportunity for fetal treatment, and we need to continue to do this research. So I'd like to acknowledge the many people in our group who have participated in this research. Really, it's led by Fetal. There are a number of people in this room who are listed in red who are part of the team. We also have a collaborative effort with Boston University. Tarek Haydar's lab are experts in embryonic brain development in various mouse models of Down syndrome, and I would like to thank all of our funders of this research. Thank you for your attention.
Thanks, Diana. We just came to the end, and thank you for staying. Just time for thanks and time for a couple of important announcements. First of all, I would like to thank to all attendees and to all the speakers, to all the contributions that you've made, and especially for the invited speakers, non-member invited speakers. Thank you for coming, and thank you for sharing us this wonderful meeting. I think we had a really great meeting. We want to thank the organizations that support us. We want to thank March of Dimes, who has always supported us. We would like to thank the commercial sponsors, our sustaining partners, our educational partners, our industry partners, and all the exhibitors for their contribution. I would like to thank all my colleagues from the board. And finally, I would like to thank the real organizers. I would like to thank very much the Scientific Program Committee, and I would like to thank CMG, Elliot, Graham, and her team work. That all the things and the beauty of this setting up is in great part due to Elliot. So I would like to just to thank Elliot very much, and all her team, that they were really very, very good. And at the end of the things, we have to recognize that Luis and Alessandro, they had made a really great job. So, and in recognition of that, I would ask Alejandro. Alessandro is here? No, it's gone. He fell asleep. No? Wait a second. I have something to do. Okay. So I really, we need to thank them, both of them, for this beautiful meeting that we had.
Great.
Luis?
Thank you. Thank you. I'll just say, I hope everybody enjoyed the meeting, and it was a chance to really try to show that the unique space that ISPD is, and to hope to attract some new members and share the meetings with you in the years to come.
Okay, I'm hoping not forgetting anybody for thanks. I have a couple of important announcements. One is that it's still open the registration for the symposium tomorrow, the symposium of the stakeholder perspective on NIPS, which is going to be held in front of this hotel at the, what's the name of the hotel? Okay, everybody knows that. So, second announcement is that we are going to have a new website almost sure at the end of this month, and a few days later, I don't know how long, we are going to have all the records of this meeting. I don't know, do we know it, Elliot, when? Oh, a few days, more than a few days, but in 8 weeks we are going to have all the records of this meeting. And the last announcement is for... The recordings, I'm sorry, the recordings. And the last announcement is for Berlin 2016, so please, I would like to invite Lynn, who is going to be the co-chair, just to show you or to say something about this. Thanks.
I don't quite know what I'm going to show you because I don't know what's on the slides, but anyway.
Yeah. Yep.
So, is that there? Yes. Yeah, so what I want to do is encourage you all to come to Berlin. Now, I've never been to Berlin, but everybody I hear who's been there say it's an absolutely fantastic place to go. We've had a brilliant meeting here this year, and we're going to have a great one again next year in this wonderful place. So please, please come to Berlin. Draft program, we're already thinking about this, so these are some ideas of what might be on the program. So think about your abstracts, think about your submissions, because we want you to come and tell us about all the stuff that you're doing between now and then. We've heard some great new stuff here this year, and we want to hear even more next year. So please, please think about what you're going to, what you want to tell us. Pre-conference courses. This Sunday we had a wonderful program of courses. But what we would like to do is we want some ideas from you. What is it you want to hear? What do you want to learn about? Have you got ideas? So can you send us ideas? Can you think of ideas, think of speakers? What is it you want? What would you most gain from a pre-congress course? So ideas from you, please. And what we will do is we're going to email you all. We've got your email addresses, and we're going to email you all with a little pro forma that you could complete and send back to us with your ideas. Because what we want to do is we want to help do what it is that you're going to find really useful. So I think that's all I really want to say, and I'm going to leave that picture up there and encourage you to come to Berlin next year. Thank you.
Okay, thank you, Lynn, and thank you, everybody. And that was it. Have a safe trip back home, and thank you again. See you in Berlin.